Welcome to AP Biology. I want to go over hydrogen bonding, van der Waals interactions, and we'll look at hydrophobic interactions. These are different forces that can hold a molecule together. So for example, if you have maybe a protein like this, it's held together by covalent bonds, but it might sort of fold and twist and turn and end up being held together sort of like this. And so it might be a hydrogen bond that's keeping this part connected to maybe the next part. So we're going to look at these forces that sort of help to hold different parts of a molecule, a bigger molecule, together. So a hydrogen bond happens when the negative part of one polar molecule is attracted to the positive part of another. So, for example, in water, here's one water molecule. And so it's got an oxygen side and a hydrogen side. So you've got O connected to H like this. So the electrons that are shared between them spend more time with oxygen. So oxygen ends up being partially negative and hydrogen ends up being partially positive because its electrons don't spend as much time there. So the bonds inside the molecule, like this, these are all covalent bonds. But the force of attraction between the partially negative side and the partially positive side right here, that makes a hydrogen bond. And so hydrogen bonds, when you add them all up together, can actually make a pretty big difference. Hydrogen bonds might form between different molecules, like different molecules of water, or they might happen in the same molecule. So for example, right here, this molecule is sort of wound a little bit, and this hydrogen on this side of the molecule is attracted to that oxygen. So that will help the molecule to bend in a certain shape. And that might be held together by hydrogen bonds. Hydrogen bonds are very weak, but altogether they can be a strong force if you have a whole bunch of them together. Here's another example where you'll find hydrogen bonds. So for example, right here, between the bases of DNA, those are hydrogen bonds that hold like the A's and the T's and the C's and the G's together. The whole rest of the molecule is all covalently bonded. All the atoms will be covalently bonded except right there in the center. And the reason is it's easier to unzip DNA that way, so that when you either want to replicate it or you want to copy it to make RNA and then the RNA could go make proteins. So either when you use it or replicate it, you would want to be able to unzip DNA. Van der Waals interactions don't come up a lot, but they're kind of cool. So you'll see this in physics class. You can have a totally neutral molecule, but if the molecule comes by something charged, maybe a positive thing over here, or maybe a positive thing over here, sorry. The electrons in the molecule might sort of zip over to the other side just a little bit and be attracted to these positives over here. And then that means maybe another molecule will be attracted by having their negative charges kind of attracted to these. And so then they'll stick because the charge distribution has become a little wacky. All right, so what's important to know from all of this is if you have something like a protein, let me just show you a protein. So let's say you've got this protein. And so this bead right here is supposed to represent an amino acid. An amino acid is, it's made out of nitrogen and carbon and oxygen and hydrogen and more hydrogen and more. Okay, so there's an amino acid and it's got an R group, which could be any number of atoms. Could be a CH3, could be a carbon with 2 more CH3s on them, could be sulfur. Anyway, there's a bunch of different R groups that this could be, but this is an amino acid. Covalent, 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 covalent. All of these are covalent bonds. We even have a double covalent bond. So this amino acid, I'm trying to represent each bead as an amino acid. And then this amino acid would bond to another and another and another. And those are all covalent bonds too. So the whole thing is covalently bonded. But what might happen is they might form these things called beta-pleated sheets, which look kind of like this. In which case, maybe the hydrogen here would be attracted to the oxygen of a different amino acid. So maybe the hydrogen that sticks off here from their nitrogen might be attracted to the oxygen that sticks off of their carbon here. And so that might happen in a few places. So you might end up getting the protein sort of attracted to the other side of the protein, just like this. Or it might form loops called alpha helices, which are a little harder to draw unless I kind of do this, right? So that would be an alpha helix. And then the whole thing might fold over on itself like this. And so this amino acid would be attracted to this amino acid. And so they're from pretty distant parts of the chain, right? But they might be attracted. They might form a covalent bond together. They might form an ionic bond, or it might be another hydrogen bond, or even sometimes a van der Waals interaction that holds them together. So all of these bonds are important in looking at protein shapes. And your proteins make so much in your body. Your enzymes are all proteins, just about. The receptors on the surfaces of your cells are mostly proteins. The markers that say, hey, I'm a liver cell, those are proteins. The steps that happen inside your cell, like there's a bunch of a cascade of reactions that can happen. Sometimes there are a bunch of enzymes that like will change something, and then another enzyme takes the product and changes it into something else, it becomes a reactant. So proteins do like I think, all the really cool stuff in your cells, and their shape gets held together by all the different types of bonds. Covalent bonds keep the amino acids in the right order, but the way it folds can be hydrogen bonds or all those other different types of bonds. All right, so this is an example of that, or an illustration, I guess I should say. So for example, here is, let's see, where's my pen? Here's an amino acid, nitrogen, carbon, carbon with an O. That's sort of the basic pattern that you have in the backbone. But then here's an R group sticking out. And notice that R group is negatively charged. But then here's one, nitrogen, carbon, carbon with an O. Maybe this protein is long and it's like going in a loop. And here's the R group, which is the part that sticks out. And notice that it's positive. So the negative here and the positive here are attracted. That's an ionic bond. And I know I said ionic bonds come apart in water, but if the protein is already folded over in a certain way, then those charges would actually help to hold the whole protein together. So that might be right here. A hydrogen bond between an oxygen and a hydrogen that may be on a nitrogen or something is right here or right here. You might also have some van der Waals interactions where the electrons here are sort of, they move over at one point and then they push the electrons here away. So you can call this van der Waals interactions. For our purposes, actually, anything that's nonpolar, you can just call these hydrophobic interactions, which means interactions, which just means that water shuns them, and water's more attracted to itself than them. And so water would just sort of push them all together because water is going to sort of be like, I don't want to hang out with you. Like water's more attracted to other waters. So the waters hanging out around here are going to be, here's a water. It's bad water. This water's like, oh my God, my hydrogen's attracted to your oxygen. And this one's like, oh my God, my oxygen's attracted to your hydrogen. So the water's attracted to itself, not to the hydrophobic stuff. So the hydrophobic stuff tends to clump together. Here's just the same kind of thing. Again, I'm showing you hydrogen bonds here, hydrophobic interactions, I'm writing it out. Hydrophobic interactions here where the water shuns them. Disulfide bond is just a covalent bond between sulfurs, that's all. And then here's an ionic bond. And I guess I liked these. Here's another ionic bond. Here's a covalent bond. Hydrophobic again. Here's a hydrogen bond. This looks like a solid line. It's actually a dotted line. You wouldn't draw a hydrogen bond as a line. Anytime you have these lines, it means covalent bonds, which mean molecules.